Hello, my name is Azizun Nessa and I'm a postdoctoral researcher based at the Centre for Endocrinology at Queen Mary University of London. I am working with Dr. Pratik Shah to identify the genetic cause of idiopathic ketotic hypoglycemia. Today, I will be speaking to you about a research project which is being funded by the Barts charity and is anticipated to begin in January 2024. The title of the project is Investigating the Molecular Basis of Idiopathic Ketotic Hypoglycemia in Children. The research will be conducted at the Centre for Endocrinology at Queen Mary, University of London. So first, a basic background of glucose metabolism and understanding what is hypoglycemia. Hypoglycemia has been defined as the lowest blood glucose concentration at which is compatible with neural function. Children presenting with hypoglycemia are at increased risk of developing neurological conditions, which can lead to permanent mental retardation or, in rare cases, even death. So what happens during hypoglycemia? The body will adapt to changes in blood glucose by releasing counter-regulatory hormones to inhibit the action of insulin and thereby raise the blood glucose concentration. Healthy children can initiate these endocrine changes, which include the secretion of glucagon, growth hormone, and cortisol and catecholamine. In childhood, the body catabolizes glycogen stores very quickly, and so the body begins to use precursors of glucose such as alanine and glycerol, which is derived from body fat and muscle tissue. To produce glucose, which produces glucose through gluconeogenesis. The lipolysis of fats generates ketone bodies such as acetoacetate, beta-hydroxybutyrate, and acetone. This provides an alternative source of energy for the brain during periods of energy deprivation. The generation and utilization of ketone bodies during hypoglycemia is a key feature in a condition known as ketotic hypoglycemia. So what is idiopathic ketotic hypoglycemia? IKH is sometimes termed as prolonged starvation. It is a diagnosis made after excluding all known causes of KH due to metabolic, hormonal, or genetic abnormalities. IKH is associated with raised ketones at the time of hypoglycemia. The age of presentation is usually from 6 months to 6 years. The incidence is unknown. However, in the United States, it is reported to be the most common cause of hospital admissions and most frequent cause of hypoglycemia beyond the neonatal period. So what are the known causes of ketotic hypoglycemia? To date, mutations in 6 genes, GYS2, PYGL, PHKA2, PHKB, PHKG2, and SLC16A1 have been identified in patients with IKH, and this may lead to various forms of glycogen storage diseases. The key feature associated with glycogen storage diseases is abnormal storage or release of glycogen. Additionally, 4 putative genes, NCOR1, IGF2BP1, SGLT2, and NEK11 have also been identified in multiple families with IKH. So what do we know briefly about these genes? The NCOR1 gene is involved in the regulation of hepatic enzymes known as phosphoenol pyruvate carboxylase 1, glucose phosphatase, catalytic, and hepcidin. The IGF2BP1 gene functions as a transcriptional regulator of several genes, including the insulin receptor. The SGLT2 codes for the sodium glucose transporter 2 protein. And lastly, the disruption of NEK11 gene has been reported to cause low glucose levels in mouse models. In our study, this is a pilot study, we aim to recruit up to 30 children with IKH, and these children have no genetic diagnosis. It is Currently not known why children and infants with IKH enter starvation mode and have an episode of hypoglycemia soon after consuming a meal. Currently, there is no explanation for this rare phenomenon of recurrent hypoglycemia with varying severity. In pathological IKH, the level of ketones in the blood does not go down, and so the children are at risk of developing major complications of chronic ketosis. So, on that account, we are undertaking this research on conditions causing non-diabetic hypoglycemia, in particular with children with IKH. The aims of our study are to gain a genetic diagnosis for patients with IKH by utilizing whole exome sequencing, to observe whether copy number variation leads to IKH, to establish the pathogenicity of variants in known candidate genes by carrying out functional studies, and to discover novel genetic causes of IKH and potentially identify new therapeutic targets. We will be using the unaffected parents or siblings as comparison, as this will aid in the identification of novel causes of IKH. Our inclusion and exclusion criteria will involve, so we will have children with documented hypoglycemia and blood glucose levels less than 3.0 millimoles per liter, with blood ketones greater than 1 millimole or urine ketones. And they must not have any underlying metabolic or endocrine causes. And so we will exclude children with underlying metabolic or endocrine causes. Children in this group have either consanguineous families or strong positive family history of hypoglycemia, diabetes mellitus, which makes it likely that they have an underlying genetic condition. The methods that we will be using is largely whole exome sequencing. Whole exome sequencing is a great tool used to identify variants in all genes at the same time. This is achieved by sequencing coding regions, conserved splice sites, and minimal promoter region areas, which contain around 85% of all known mutations. Whole exome sequencing has been proven to minimize the time taken from disease presentation to disease diagnosis, hence reducing the overall cost of diagnosis and increasing the diagnostic yield. In addition, the technique has also been successfully applied in numerous studies. We will be sending our DNA samples to an external service provider that will carry out the whole exome sequencing. The first stage of analysis will involve screening known genes that cause IKH, followed by searching for potential new genes. The sequencing data of patients who have no mutations in known genes will be explored further to seek out novel genetic causes and potentially identify new therapeutic targets. In silico prediction software will be used to predict the pathogenicity of variants in putative genes. This figure is a representation of what we expect to find within the 30 patients based on previous studies. Brown and colleagues had a large cohort of 164 patients, of which 24 were given a genetic diagnosis. So we expect around 12%, which is approximately 4 of our patients, to have a mutation within the glycogen storage genes. Based on the findings of Al-Hayden, we expect around 12 out of 30, so 40% of patients, to have a genetic diagnosis in the candidate genes. This leaves around 14 patients where we do not expect to find any known causes. And on these patients, we will recruit the parents and unaffected siblings and do trio exome sequencing to identify rare variants. Once we have completed whole exome sequencing on all 30 patients, we will proceed with Sanger sequencing to confirm that the mutations are present in the patient, parent, and siblings. And eventually, we will do functional assays on the variants that we have identified. Overall, this research will hopefully unveil new causes of IKH, which is a heterogeneous disease. Currently, only half the cases of IKH have a genetic diagnosis in one of the 10 genes, and up to 50% of cases have an unknown cause. With no testing available on the NHS, we believe whole exome sequencing is the best option to identify defects not only in gene known to cause IKH, but also to discover novel causes and mechanisms which may lead to better treatments if the mechanism is understood. Using this as a pilot proof of principle study, this will help us to expand to a larger study of IKH worldwide with patients recruited through our networks. This work will provide the foundation for grants to understand mechanisms of novel genes and investigate treatment with pharmaceutical companies to generate novel drugs and improve patient outcomes. I would like to give thanks to all our colleagues at Barts Health NHS Trust, as well as the QMEL team, which is Professor Lou Mathirel and her lab team. I would also like to thank Ketotic Hypoglycemia International for giving me this opportunity to speak at this conference. Thank you. If you have any questions, please email me or Dr. Pratik Shah, and we'll be happy to answer your questions. Thank you.